Anthony Green, who comes to us from the University of Manchester, received his PhD in synthetic organic chemistry under the supervision of Professor Thomas. He then went on and did a postdoc research with Professor Turner and Flish, based in Manchester Institute of Biotechnology. Following his postdoc there, he did a second postdoc in Zurich with Professor Hilbert. Recently started his independent research in 2016 at Manchester Institute of Biotechnology, where he's a professor of organic and biological chemistry. And his research interests lie in the design and evolution of enzymes with new function and the use of genetic expansion. Take it away, Anthony.
Great. I hope everyone can hear me okay. So first of all, thanks for the very kind introduction, John. Also like to thank all the organizers, some old friends of mine as well. So it's really good to see some of those guys in person again. And so today I've been very much a user of GCE technology rather than a developer of the technology. And so I'm just going to tell you about our efforts today to build enzymes with new functions. And so this is kind of just like a summary and an overview slide of some of the activities that my group are involved in. So in the broadest sense, we're interested in basically creating enzymes from scratch for a whole range of different applications. So in order to achieve this, we use like a range of techniques. So I guess advanced active site design and engineering techniques. Particularly relevant today is we attempt to install new catalytic elements into proteins, and that's particularly relevant to the discussion of the topic under discussion today. We sometimes repurpose natural scaffolds, but sometimes we build entirely new scaffolds, and so some proteins that are, there's protein folds that are not found in nature. We extensively rely on directed evolution to tune the efficiency and selectivity of our systems. And we use a whole, not only structural biology, but also biophysical and spectroscopic characterization to understand how our enzymes operate at the molecular level. With the ultimate aim there really is so one day we might actually just be able to design these things from scratch rather than having to rely on high throughput experimentation. And here are just some examples at the bottom of the screen of enzymes that we've worked on over the years. Okay, so I'm just going to, before I get onto the genetic code expansion element, I'm just going to sort of build in with a link to biocatalysis. And so the field of biocatalysis typically is like almost exclusively involves the top-down engineering of natural enzymes. And so my lab do maybe about 20% of my lab work in this area, typically when there are big application areas to work on. And so this is a project that we did in collaboration with the funded by the Bill and Melinda Gates Foundation that we did in collaboration with Nick Turner's lab, where we engineered a cytidine deaminase, which is a natural hydrolytic protein. And we re-engineered this protein so that it could accept hydroxylamine rather than water as the nucleophile. This let us produce this key intermediate, this N-hydroxycytidine, which is a key intermediate of molnupiravir, which is one of the few approved COVID-19 antivirals. And we could this enzyme is actually really good. So it could do many hundreds of thousands of turnovers. So we could scale this up to kilogram level scale. And this is now being exploited by sort of manufacturers to produce this therapy. And so that's how the majority of the time we access enzymes with new functions. But in some cases, nature just hasn't given us the mechanistic repertoire for certain important transformations. And so if we just go one step beyond that, then what do we do in a scenario where there is just no good starting point to do enzyme engineering? And so, I'm kind of going to go quite quickly through this example. So what we can turn to here is computational enzyme design. And so there's a reaction, which is a very famous reaction in organic synthesis called the Morita-Baylis Hillman reaction. This is a CC bond forming enzyme where we couple two units together, such as compounds 1 and 2, so an alpha beta unsaturated carbonyl and an aldehyde. And we can couple those together to get a new CC bond formed, generating a new chiral center. And I say this is kind of one of those named reactions that are used frequently in organic synthesis, but it's a really notoriously slow reaction. So typically it takes many days to do this reaction. It's typical to drive reaction to completion. And so we were interested in developing an enzyme for this reaction, but again, this is just a reaction that's not observed in nature. And so using computational enzyme design, I guess I'll talk through this relatively quickly because it's not related to genetic code expansion at this stage. What we've got is an enzyme that uses a histidine nucleophile, which is going to do a microaddition to this alpha unsaturated carbonyl. And we've got several oxyanion stabilizing units, this glutamine 128, this serine 22, which is basically effectively ordering a water molecule, which stabilizes these two oxyanion intermediates. So this was a computational design from David Baker's lab. Most computationally designed enzymes are extremely poor, and in this case, this was extremely poor is probably an unfair characterization. So the efficiencies of those enzymes are orders of magnitude away from natural enzymes. But what they do serve as is really valuable starting points for engineering, so we can start to engineer those enzymes to have efficiencies that are more akin to natural enzymes. And so we did that exactly that thing in this case. We started with this computational design enzymes, and we could the power of enzyme engineering is we can subject this to directed evolution and massively enhance the efficiency of these systems. And so over 40 rounds of evolution, what we can see here is we installed, I think it was in the region of 22, 24 mutations into the computationally designed protein. And you can see here that you can just gradually improve not only the efficiency of the reaction, but also the enantiomer selectivity. And so the final variant we characterized was 700-fold more active than the starting variant. And you can also see that we've also evolved high levels of stereo control in this system. And here's just some structural characterization of how our enzyme operates. And what was really interesting, if we remember, how we tried to design this mechanism. So, you have to remember that we don't have a blueprint to work from, so we can't just look at natural proteins for how they promote the reaction, because there is simply no natural protein for the reaction. So we're relying a lot on chemical intuition. And when we evolve the proteins, often interesting things happen in the active site, so you can get mechanistic solutions that you don't necessarily expect to happen. And what happened in this case, basically, is that most of the octane stabilizing residues were abandoned during evolution, and instead this arginine 1,2,4 residue emerged. And this arginine can sort of move between conformational states and can serve as a really elegant way of stabilizing multiple oxide intermediates, which are formed on this complicated reaction coordinate. And you can just see from this table of data here that if you take out the arginine, you almost completely lose activity in the protein. And so basically, we've been able to engineer bifunctional catalysis into this protein. Okay, so now I am going to come on to the genetic code expansion element. Okay, so what I hope I've shown you initially very briefly is that we can now, using computational design methods, we can build new active site environments in proteins. We can embed those active sites into either de novo scaffolds or natural scaffolds, combine that with evolution, and get relatively proficient enzymes, certainly enzymes that are orders of magnitude more efficient than small molecules. I guess the challenge is when we, as an enzyme designer, what I always feel was always a bit uncomfortable with is the fact that we're really restricted with what range of active sites we can build into proteins, because we only have a limited range of functional groups that the genetic code allows us to introduce. Okay, and so basically, when I started my group, I wanted to try and resolve this issue and basically start to build active sites out of a far greater range of functional amino acids. And I think, being a GCE seminar, it's probably obvious like where this is going to go, is that what we elected to do was build enzyme active sites out of new catalytic elements that are simply put into proteins as non-canonical amino acid side chains. And to do this, we can engineer cellular translation components in order to specify our amino acid of interest. We typically encode these in response to the TAG stop codon. And because we encode these new functional elements on a genetic level, we can still take advantage of directed evolution to further enhance and augment the functions of those proteins. And so I've just shown one example here, this N-methyl histidine, and I'll give an example of where we've used that in a second, but this has been like a really useful and valuable tool for us in enzyme engineering. So we've been able to use this to modulate catalysis by metalline cofactors in proteins, or alternatively, it serves as a really valuable catalytic nucleophile. And we have several published work in this area, but we've been able to build enzymes that operate through mechanisms that are inspired by small molecule organic catalysis, where we use this N-methyl histidine as a catalytic nucleophile. And I'm just going to present one example of this where this has been really useful. And so one of the really challenging, it's kind of one of the fundamental reactions in biology is hydrolysis. So ester hydrolysis is a really fundamental reaction. There are many natural proteins, of course, which do hydrolytic reactions, often using catalytic triads and catalytic nucleophile which are embedded within catalytic triads. But from a design perspective, this has been one of the real great challenges. Okay, we so far haven't been able to replicate catalytic, to get a functional hydrolase has proven really challenging. And the reason why is that we've been able to design a whole range of, we can accurately embed nucleophilic residues into proteins. And so we can develop proteins that have got really reactive histidine or serine or cysteine nucleophiles and get to these acyl enzyme intermediates. The problem in all cases so far, though, is that it's simply not possible. It's not been possible to turn over these intermediates. And so the energy demands on this second step have been too high. And basically what we propose is happening is that we're forming these neutral acyl enzyme intermediates, and these are just really resistant to hydrolysis. I've just shown a few examples here of publications where where, as I say, we've basically been where people have been able to accurately embed these nucleophiles, but you just simply can't get any hydrolytic turnover. And we felt that we could apply genetic code expansion technology, taking inspiration from work in organic synthesis, to address this challenge. And I just want to quickly explain how we did that. Okay, and so we're going back to this enzyme, this protein BH32, which was the computationally designed Baylis-Hillman catalyst that I talked about earlier in the slide. Okay, so this catalyst is designed to use a histidine 23 nucleophile, and in blue here you just have the computationally designed residues around the active site. Okay, and so what we found quite early on was that this enzyme was also a promiscuous hydrolase. Okay, and if you look at the reaction profile of this, so if you look at the reaction profile, what you can see is you get this burst behavior, which correlates to the release of fluorescent. Okay, so we're going to use an activated ester, so this fluorescent ester is the substrate. And what you can see is you get this burst behavior, which correlates to one turnover. So what that's reporting on is the formation of this acyl enzyme intermediate. But you can see the reaction then is effectively flatlined. Okay, and you can see here you've got this low background reaction. So basically, what this means, this is your classic burst behavior that happens within all design hydrolases. Okay, so you're not really doing even a full catalytic cycle, you're just getting stuck at that intermediate stage. Okay, and we proposed that the reason was basically is that upon addition of the histidine nucleophile, you can lose the proton on the non-coordinated nitrogen and generate this neutral acyl imidazole complex. And it's known in organic synthesis that if you make acyl imidazoles, you could even put them through a silica column. So they're quite resistant to hydrolysis. And so our proposal was that, first of all, we have to know, our proposal was that if we just simply methylate that nitrogen, so supply N-methyl histidine rather than histidine, we should be able to generate N-acyl imidazolium complexes. And so in order to do this, the first thing we had to know is which of the two nitrogens was reacting. It was designed to react through the N-epsilon, and that we've got a crystal structure here showing that that's actually the case. So if you treat this with an alkylating agent, you can see you can get reactions through the N-epsilon. What that means is that we have to encode N-delta methyl histidine. And thankfully for us, Peter Schultz's lab had already developed a synthetase which could encode methylated histidine into proteins. And so we could simply switch out the histidine 23 by methylated histidine. The idea here is you then generate this charged acyl imidazolium species. It should be therefore a superior catalytic nucleophile. And that's what we can start to see. So this was the first time that we were seeing any catalytic turnover in a designed hydrolase. So you can see here that the kinetics have now become linear in nature, although albeit it's a little bit slower than the burst with histidine, but we can correct that through evolution, which I'll show in a minute. You can see now that you don't have that biphasic behavior and you can start to get multiple turnovers. A second thing that was important is that we combined for us, and when we first started there was very few reports of doing directed evolution in the context of an expanded genetic code. Okay, and so we've developed over a number of years. Like we've got a number of programs where this is perfectly feasible, much like any other evolution project. And so what we can do now is start to cycle this enzyme through rounds of directed evolution. I won't go into too much detail, but you can see the early rounds were difficult because you're with low activity and low expression, you're fighting against cellular background, but you can see that then you can start to really make quite substantial gains in directed evolution. And I think here we stopped at, because this was like kind of a proof of concept study for us, we stopped by introducing, I think, 7 or 8 mutations into the protein. And now you can start to see that you can get appreciable levels of catalytic turnover. We have all the kinetic characterization where you can see we've got a 15-fold increase in catalytic efficiency. This is dominated by an increase in K-cat term, which is typically the more difficult parameter to engineer into proteins. And as I say, this for us was a really proof of concept. We could have pushed this much further from an evolutionary perspective, but we're just showing now that you can do you can basically build new mechanisms. You can solve a long-standing challenge in ester hydrolysis design. And just found the experiment which we were a bit nervous about was replacing the N-methyl histidine by histidine after the evolution. But you can see that the catalysis is strictly dependent on this on the N-methyl histidine catalytic nucleophile. And just finally, something that's important to note is that the enzyme is about 9,000-fold more efficient than methyl histidine in free solution. And so I think that for us shows the benefit of being able to introduce these catalytic elements into protein cavities and then combine that with evolution, where you can still, and so basically you can just get efficiency gains that you simply can't get in any other manner. We almost always structurally characterize our proteins, and in this case it's interesting because what you can start to see now is what catalytic mechanisms emerge as you put these, as you optimize efficiency. You can start to understand what are the molecular features which emerge that correlate with that improving activity. And so one thing we can see here from the active sites perspective is that you've got this asparagine 46 residue emerged during evolution. This coordinates two ordered water molecules in the proximity to the N-methyl histidine. And although we don't have the full detail analysis, these waters are almost certainly involved either in important proton transfer steps, or one of them may even serve as the catalytic water nucleophile in the reaction. A mutation that was interesting was one that was a little bit more remote from the active site. And so what you've got here is this is a two-domain protein. So you've got a cat domain, which is where the catalytic residue is built into in this core domain. And this mutation was quite remote from the catalytic center, but it was right at the interface of these cap and core domains, this leucine to proline mutation. And what we can see is that if we use a mechanistic inhibitor, what we can see is a global change, sort of a global domain motion. Okay, so we can see that the cap and the core domain in isolation overlay quite nicely, but when you inhibit the protein, what you find is that you've got this like hinge motion which opens up. And what's interesting is that if you go back right to the natural activity of this protein before the computational design, that protein also relies on domain motions which are linked to the reaction coordinate. And so, although a lot of work has to be done to prove this is the case in this case, what we believe we converged on here was functionally relevant protein dynamics. And I think this is kind of one of the first examples where we can see that those in a designed enzyme where global domain motions actually contribute to catalytic function. So we think that's kind of an interesting point that we're following up on. Okay, so Bill, we took advantage of this. We're just going to present one slide before I want to move on to the final topic that I'm going to present today. Back to our Baylis-Hillman story. In principle, methyl histidine should be a superior catalytic nucleophile than histidine for this kind of transformation. And so what we did is we basically embedded N-methyl histidine at one of our intermediate points in evolution from our earlier study. And we then took that protein on a different evolutionary journey. Okay, so we basically were purely screening for phenotype. We basically got a 920-fold increase. So this is all now being done in the context of having an N-methyl histidine nucleophile. So what we land on here is a protein which ultimately is over an order of magnitude more efficient than our earlier design Baylis-Hillman. So this is actually now quite an efficient protein. But what I think is more interesting is that although this was in no way deliberate, this was purely just applying phenotypic screening. What we converge on in this case is a completely different catalytic mechanism. And what happened is that the arginine, which was so critical to function in our earlier BH32.14, which works in synergy with a histidine residue, was abandoned during evolution. And in the context of an N-methyl histidine residue, it's actually a glutamate 26, which emerged during evolution, which plays a really crucial role in catalysis. So this basically told us that if you even making really subtle modifications to that key catalytic element and taking proteins on different sort of divergent evolution journeys, what you get actually is quite distinctive mechanistic solutions out of those processes. And this is really one of the first times you can't typically do this within the constraints of the genetic code. This is, to my knowledge, the first time that you've seen divergent evolution where the only change you've made in the protein is a single methylation event of the key catalytic residue. So we're sort of writing this work of publication at the moment. Okay, so I'm hoping John shouts out if I've got 5 or 10 minutes left. Okay, so what I want to tell you about now is a really recent study from my lab where we start to now build in catalytic mechanisms that are just simply not possible to access within the constraints of the genetic code. Okay, and so what I want to show now is our efforts to design enzymes or photoenzymes that operate through a mode of catalysis known as energy transfer photocatalysis. So this is a really important mode of reactivity in organic synthesis. I'm going to explain how that works in a second, but it's just simply not accessible as a mode of chemistry in proteins. And hopefully I'll explain why this is. Okay, and so what catalysis relies on is this unit called a photosensitizer. Okay, so this is typically an aromatic ketone. And the way it works, upon irradiation with light energy, this sensitizer very efficiently populates this long-lived T1 excited state. In the presence of a neighboring substrate then, and in solution, this is just through collisional events, this triplet energy can be transferred to the substrate. In a spin-allowed process, it transfers the substrate to an excited T1 state, and this is then sufficiently long-lived to undergo a wide variety of either intra or intermolecular processes. Okay, and so basically, say that lets, in organic synthesis, you can access a wide range of reactions by taking advantage of this triplet energy transfer process. And one of probably the most famous reaction is this 2+2 cycloaddition, which from the Woodward-Hoffman rules is predicted to be a thermal equivalent reaction, and that is actually the case. Okay, so you need light energy to promote that reaction, which contrasts with the thermally allowed 4+2 cycloaddition reactions. And it's thermally forbidden because you have these incompatible orbital symmetries in the ground state. Okay, so the major challenge for photocatalysis is how do you control reaction selectivity? It's actually really challenging. And so there are a small number of chiral photocatalysts which are known, but they only really work on a very narrow range of substrates. Okay, and I haven't got time to talk in great detail about these today, but I just want to show you what we felt was possibly a more complete solution. Was, are we now in a position to use protein scaffolds to mediate an anti-selective transformations? Okay, so then we can rely on the versatility and the engineerability of protein scaffolds. And because we now have access to this expanded genetic code technology, we should now in principle be able to encode suitable photosensitizers to mediate triple energy transfer processes. Okay, and so the idea is that we encode units such as benzophenone into protein scaffolds. We combine it with directed evolution, and in this way we can really maximize productive interactions between the sensitizer, the protein, and the substrate, ultimately giving us highly proficient and highly anti-selective photocatalysts. So that's the ambition and the goal. Okay, and so basically what we started off with, because of mechanistic similarities between Diels-Alder reactions, which are 4+2 cycloadditions, and 2+2 cycloadditions, we took this computationally designed Diels-Alderase, BA20. So this was originally designed by David Baker's lab, subsequently Don Hilbert's lab converted this into quite a proficient Diels-Alderase. And then we're going to try and repurpose that into a 2+2 cyclase. Okay, so we embedded the sensitizer at position 173. This, you can see here that you still have sufficient room for substrate binding, okay? And this tyrosine and glutamine residue will become important to the story later on. And we targeted this intramolecular 2+2 reaction. And so if you do this reaction just with UV light in the absence of any catalyst, or if you use it, sorry, if you use this with a non-chiral sensitizer such as benzophenone, what you get is about a 50-50 mixture. It's actually about, I think, 55 to 45 mixture of the straight product and the cross product. And of course, in the absence of any chiral elements, you produce these as racemic mixtures. Okay, and so here's catalysis with benzophenone. You can see in purple is the straight, in yellow is the cross, and you can see here the reaction conversion, the defined reaction condition is in the region of 8%. And what we found is that if we put this sensitizer at position 173, this is where we become interested as enzyme engineers, because two things are happening. Okay, first of all, we're increasing the reaction conversion. But more importantly, we're starting to get some modest levels of stereo control in this reaction. And what that means is that we have some evidence of protein catalysis taking place. Okay, so that for us then is just our starting point to engineer the enzyme. Okay, and so we had to develop in this case directed evolution workflows that were suitable for light activated processes. Okay, so this took us a little bit of time because the extra step you need is you need to go through uniform irradiation across 96 well plates, which is not so trivial. And the goal of this project is to engineer the protein just to produce a unique product. Okay, so we targeted this product 1A, so this is the straight chain isomer as a single enantiomer. So that was the goal of the project. Okay, and we can see that we can then, so here's the activity with benzophenone. We've got conversion on the y-axis. We've got an enantiomer excess on the x-axis. Here you can see our first variant that we've already told you about, where you've got this modest EE and modest improvements in conversion versus benzophenone. But if we start placing mutations into the protein now, after screening bioevolution, we can get dramatic improvements both in reaction conversions, and also we can get 100% reaction selectivity from this process. Okay, just to show you what these reactions look like over time, if you go to our N1.0 catalyst, okay, so this is the catalyst shown here, you can see that you get this initial rate of reaction when the reaction stops. Okay, so the catalyst is undergoing deactivation. We can talk about that later if anyone has any questions about that. But after evolution, now we can get full conversion, at relatively low catalyst loading. So the enzyme is able to do about 400 turnovers. So still some way to go to make it into a true enzyme, but you can see that you can really get dramatic improvements. You can also improve, you're also here improving the initial rates of reaction as well. Two features I want to point out about why these enzymes are so interesting as well is that, first of all, they operate at room temperature. And this contrasts with any other small molecule photocatalyst, which typically requires cryogenic temperatures to get it to work. The second aspect is that the reactions are completely tolerant of oxygen. Okay, so normally photocatalysts are very efficiently quenched by, they undergo triplet quenching of oxygen. In our case, our enzyme is able to protect, I guess, the cofactor from catalysis from oxygen, possibly by excluding oxygen from the active site. We've got to do more work to understand that. Yeah, we've got these completely aerobic tolerant enzymes which are able to operate under ambient conditions with exceptional levels of stereo control. Okay, so we were able to do a range of okay, should I wrap up now, John?
It's like 2 minutes. You have 5 minutes left total.
Perfect. Two minutes is good. Okay, so we can work on a range of substrates. I'm just going to highlight these two here because these are really difficult to do with small molecule systems, because the cyclization step is very slow. And what that means is that what happens with small molecules is you generate the excited state complex, but then it just dissociates from the catalyst and you have non-selective chemistry going on. Our proteins are able to resolve those problems. Okay, so you can start to access classes of substrates you can't normally access. Even though we've only put this through a little bit of evolution, the active site is actually quite sophisticated. And so what we can see here is, and I don't have time to probably talk in too much detail, you've got a really nice alignment between the benzophenone cofactor and this is a product actually bound in the active site. But what you've also got is really nice hydrogen bonding networks, which are activating this substrate for catalysis by lowering the triplet energy. We can then start to carve out the active site to perform more challenging transformations, for example, where you've got alkylated quinolone type substrates, and also quite cool is we're not limited to intramolecular processes. So we can also do bimolecular processes with extremely high levels of stereo control. And this is really just the start for us. Like this is, photocatalysis, there are literally thousands of photocatalytic reactions that just can't be promoted in any anti-selective fashion. And so we can now tap into all that and start to build enzymes for these useful photocatalytic processes. And so I'm just I think probably I don't need to talk in detail about the summary. I'll probably remember what we've talked about. But basically the summary is that genetic code expansion has been just an invaluable tool for my lab in enzyme engineering. And I say, it's nice. I get to like meet and work with the genetic code expansion developers. We're very much on the utilization of the tools, but I think it's an amazing research field and it's been extremely valuable to us as a research group. And I should just finally acknowledge all the amazing students that we have. We have like really great postdocs, great students in the lab. They, of course, do all of the work. They also have the vast majority of the ideas, I should say, as well. And they're really like a pleasure to work with. I also have great collaborators, just a few of them mentioned on the slides here from projects that we've done together that we've actually talked about today, but we have lots of great collaborators all over the world. Thank all the funding agencies, and yeah, thanks everyone for listening to me.
Great.
I hope
you
have
probably 2 minutes left. But no, that was really great. So we can take questions, a couple questions, short questions, by raising your hand, Q&A. I'll start off. So how do you reconcile using genetic code expansion with the loss in efficiency of generating these enzymes with the overall change in catalysis efficiency that you get? So do you just scale up to accommodate the inability to make, because it's inefficient to suppress stop codons to generate these enzymes? And so what's your mode to get around that?
Yeah, so it depends, like, how efficient the translation components are, of course, is probably the biggest driver on how much protein yield you lose. And so we can just make components more efficient. That's one thing. In terms of, at the moment, I think the biggest thing for me is like, using this for an application which you simply can't access otherwise. So the photochemistry is that example where there just simply is no way to do that triple catalysis in proteins any other way. There is then this element of, okay, if we're ever going to use that in anger on any kind of scalable process, we then have to, but that's the thing. But I think the first thing is we had to show we could do something interesting that people want to scale and that you just can't do any other way. And then it's a question of working with people, working with our industrial partners, for example, to say, okay, well, what do we now need to go and do to make this actually a scalable technology? I think that's kind of the second step for us now. Great.
So I have a question from Arush. I'll allow you to talk if you'd like to ask the question.
Hi, am I audible?
Yep.
All right. Thank you so much. That was a very interesting talk. So I've just got a question about the last 2+2 cycloaddition you showed, because I know Thorsten Park was working on a very similar reaction, and he was just using benzophenone, like modified benzophenone as a catalyst. And as you said, that reaction worked at -25 degrees or something. So what do you think is the reason that with an enzyme, you can get it to work at near room temperature conditions?
Yeah, so Yeah, so basically, so first I should say that I know Thorsten very well, and his work is obviously the inspiration for the design of those enzymes. So I think the reason what you have is that as you go to low temperatures, you take out some of the when you generate a triplet state, you have non-radiative quenching of the triplet state through vibrational modes and also from some collision modes with solvents and things. And as you drop the temperature of the reaction, so what you've got going on, you've got basically these non-productive processes, and then you've got productive energy transfer. As you drop the temperature, you change the partitioning of those two processes. So you start to reduce that non-radiative decay process, which is basically slowing everything down. But you don't change the energy transfer rate by such a degree, if that makes sense. And so it basically that's the main thing, yeah. So what the enzyme can do is, what we know is that if we put anything into the active site, so if we lock the product into the active site, which is even the crystal structure, we massively extend the triplet lifetime. And so basically by locking the product in, you basically almost certainly what you're doing is basically stopping protein motions, which are quenching your triplet through non-productive modes. So it's probably not a complete answer because I don't have all of the data yet. We've still got fire in the data, but I hope that gets us halfway towards the answering your question.
Okay, yeah, thank you so much.